Let us now understand another phenomena, that is photorespiration. It is also known as oxidative photosynthetic cycle or C2 cycle. Now, to understand photorespiration, we have to recall the first step of the Kelvin cycle. Over here, the RUBP, that is the ribulose biphosphate, combines with combines with carbon dioxide in presence of enzyme Rubisco to form two molecules of 3-PGA. So here we are forming two molecules of 3-PGA in case of C3 plants. Rubisco enzyme is actually peculiar, and it is, that is the ribulose or carboxylase oxygenase. It is peculiar because this enzyme has active sites which can bind with both oxygen as well as the carbon dioxide. Now, the Rubisco enzyme is known to have more affinity for carbon dioxide. So, if we see Rubisco enzyme, under normal condition this will happen, but it has more affinity for carbon dioxide, and at the same time it can bind with oxygen also. Therefore, the name carboxylase oxygenase. When it combines with carbon dioxide, it is carboxylase. When it is combining, or when it is helping in combining with oxygen, it is oxygenase. So over here, since it is helping in combining with carbon dioxide, it is carboxylase. Rubisco enzyme is functioning as a carboxylase. Now, this active site binding is competitive in nature. So that means the concentration of oxygen and carbon dioxide will decide whether which one will bind with the enzyme. Now, this enzyme Rubisco is also the most abundantly found enzyme in the world. And when we talk of the C3 plants, some of the oxygen, it binds with the Rubisco enzyme. So if we see over here, the RUBP has active site. RUBP combines with, both are available. So Rubisco over here and oxygen and carbon dioxide. So in C3 cycle, some of the oxygen combines with RUBP with the help of Rubisco enzyme. And as a result, instead of forming two molecules of PGA, it will form one molecule of phosphoglycolate. So it will form, if it combines with oxygen, it will form two molecules of phosphoglycolate. This we are talking of C3 plants. Try and understand it again. Carbon dioxide, it will form 3-phosphoglycerate. Again, try and understand. Rubisco enzyme can function or can combine with carbon dioxide as well as oxygen, but this binding is competitive in nature. That means depending upon the concentration of carbon dioxide or the temperature or different situations, it can vary its nature. That means it can combine with oxygen or it can combine with carbon dioxide. So in C3 plants, sometimes the RUBP is made to combine with oxygen. That means Rubisco is functioning as oxygenase over here. And as a result, two molecules of phosphoglycolate are produced. This phosphoglycolate undergoes a process of photorespiration. And a molecule of carbon dioxide is released. This process requires ATP in the form of energy. And this phosphoglycerate then enters into the Kelvin cycle, releasing oxygen as a byproduct, and RUBP is restored again. If we understand this phosphoglycolate molecule, this is a 2-carbon atom compound. Therefore, the name C2 cycle. Try and understand it again. In certain conditions, the Rubisco enzyme make the RUBP combine with oxygen, and otherwise it is normally combining with, most of the it is combining with carbon dioxide. So RUBP, when combining with oxygen, it will form 2 molecules of phosphoglycolate, and with carbon dioxide, Rubisco enzyme helps in formation of 3 PGA. That is the normal. Now if it is combining with oxygen forming phosphoglycolate 2 molecules, this phosphoglycolate molecule undergoes the process of photorespiration by a series, and here the carbon dioxide is released as a byproduct, or is released over there utilizing the energy of ATP. This phosphoglycolate by the process of photorespiration goes to form 3-phosphoglycerate, and which finally enters into the Kelvin cycle. But here, since it is using ATP and releasing carbon dioxide, a lot of energy of the plant is wasted, and as a result the carbon dioxide fixation is reduced. Therefore, photorespiration helps in reducing, or it is a wasteful process because it is not helping in the synthesis of the sugar. Instead, it is releasing carbon dioxide and it is utilizing ATP. So it is a completely wasteful product. When we talk of the C4 plants, there is no photorespiration taking place. No photorespiration is taking place because they have a mechanism by which they increase the concentration of carbon dioxide at the enzyme site. Why? Because the carbon dioxide is taken up by the PEP, and it is from the mesophyll cells, it is broken down and is passed on to the bundle sheath cell. So as a result, the Rubisco enzyme function as a carboxylase, and in the case of C4 plants, it does not act as an oxygenase. So therefore, C4 plants are more efficient in carbon dioxide fixation as compared to C3 plants, because of the fact that they do not have the process of photorespiration. And that's why the productivity is found to be greater in the case of C4 plants. Photorespiration is a wasteful phenomena which is taking place in the C3 plants. This is one of the major differences between the C3 and the C4 plants.